Hi, this is me Vishal again. This is my foremost video of trial 2. I'm going to make the soil solution, like I'm going to put the soil into the tap water and then swap it into the agar plates. Last time we used the dirty plates, but the microorganism would not appear properly. That's why we are using that soil and the tap water for the betterment of the result. Here is my agar plate ready. This is a 80 ml isopropyl, 70 ml, 60 ml, market made, without active ingredient, water, and my swabbing stick is ready. Controls, that are my 6 controls. This is the solution with the that I added soil into the tap water, and then I'm going to swab it on the agar plates, then pour the 2 drops of every control and see the result after 48 hours at the room temperature. So I'm going to swab it on and let's see the result after 48 hours. This is my plate. I just step the holes on it, and every 6 quadrants, every 6 controls, and I swab it the soil water solution on it, and these are the controls. And I am expecting the colonies on the water side or on the without active ingredient side, or maybe on the 60 ml isopropyl alcohol side. And for the 80, 70 and the market made, I'm not expecting the colonies because isopropyl is the independent variable, and the zones that create on the agar plates are the dependent variable. So in these 3 quadrants, the alcohol concentration is very high, so I'm not expecting the zones or any holes or colonies on that quadrant. Hi, so I'm taking the result today. So this is like 70 mil, 80 mil, and my market made. You like my result is not all like properly accurate, but the 70, 80 and market made there's less colonies. There's like nothing grows on these 3 quadrants, but if you can see to these 4 quadrants, there's a growth of the microorganism. You see this one, so this is the 60. There's a growth on 60, and on the H2O side there's also growth here, and the without active ingredient there's a growth on the without active ingredient, and this is the market made, there's like no growth. And for the 80 ml there's only one like little dot, but this is under the punching thing, and there's a 70 ml, there's nothing here. So 60 ml there's a growth on the 60 ml side, but H2O side there's a growth without active ingredient. So there's a growth on the 4 quadrant except 80, market made, and 70. So my final result is almost complete. So this is the last video. Thanks.
Hi, my name is Gajeena. My section number is 104 for microbiology project BI 206. My partner is Vish Patel, and my topic is called hand sanitizer production and testing efficiency on microorganisms in a dirty plate. The research questions are: can microorganisms be destroyed without the help of active ingredients in hand sanitizer? What is the difference between homemade hand sanitizer and store-bought hand sanitizer? And how many volumes of hand sanitizer can be applied in the hands to prevent bacteria from spreading? So far, the 3 hypotheses are: only store-bought hand sanitizer and homemade hand sanitizer with active ingredients 70% isotopic ethanol and hydrogen peroxide can destroy the microorganism. The hand sanitizer result can destroy bacteria and foodborne diseases compared to hand washing with soap and water, and only 2 milliliters of hand sanitizer with active ingredients can prevent contamination caused by microorganisms from a plate that's dirty. So far, here's the result. This plate. This one only it took me 2 weeks of waiting from I started it on week 7, Wednesday, I believe, and I found out that this is my result. There's only these 4 colonies. They are contamination. But this, in the section of 60% alcohol, is Penicillium fungus. It's the only one that appeared. The 70% and 8%, I believe that I did not give enough samples, or maybe because the sample from a dirty plate is not effective enough. And for this one, that contains not the alcohol, only this plate that contains 80% alcohol, 70% alcohol, and 80% alcohol. This plate contains store-bought hand sanitizer, water, and water plus soap. This one, with the store-bought hand sanitizer, got my organism, Staphylococcus aureus. They are clutched together. It's creamy and it's elevated together. The colonies you see are browned, and the surface is, the inside is curly. Penicillium fungus is round and it's elevated. So right now I'm doing trial 2, and instead of using a dirty plate, I'm going to use saline. Saline is made of salt and water, and I'm going to add it to my hands and use a swab to swab it. Okay. The environment is going to be, for the experiment, is the agar plate, which is obvious, and it's going to be in the room temperature. The independent variable is going to be the amount of alcohol, and a dependent variable is going to be the wealth. We'll see, the wells contains the colonies that has the well have going to be produced. The well is going to be used to the size of it will be used for the alcohol to be stored, so that the colonies or the bacteria can be slowly, not grow. Okay, here's are the materials. This one is 60% alcohol homemade hand sanitizer, 70% of alcohol homemade hand sanitizer, 80% of alcohol, homemade hand sanitizer, store-bought hand sanitizer, and a cup of water plus soap are these 5 are the positive controls, and the water is the negative control. Now let my experiment begin. Now I'm going to dip my hands in saline. I know I showed you the video of the cup with saline, but it does not be effective enough, so I decided to pour it into a bowl. See, now I've been dipping my hands for a minute now, and okay, that's good. Now time for the swab. It's not. Maybe we need more. Just hold on a sec. Okay, let's do this. Okay, let's see. Just gonna put it here. Okay, I'm going to dip my hands in saline. I already did it for about a minute. Now I'm going to do the swabbing. Now time for me to do the spread plate. Okay. That should do it. Okay, I got my bulb, and I'm going to use this. I already labeled this plate. Yeah. 22 Celsius is the room temperature. I'm going to do it. Okay, I'm going to begin with water. This should do well. Nope, not enough. 1 liter could do a trick. See, 1 milliliter of water. Now let's begin the welling. Going to stab here. Okay, do it slightly. doing it. Oof. Okay. That's a good successful boiling. Only some small amount of water enters inside the agar. That's good. Now I'm going to do water plus soap for my second well. Okay, that's good. Second well is going to be here. Yep. Okay. Easy. Yep. It's a success. Now I'm gonna make the third and final well in this agar. Store-bought hand sanitizer. Yep, I did it. There. This is successful. Now I'm gonna wait for it to be put in a dark place. Or not. Like in trial 1, it took me 3 weeks for the group to come without, like, in a dark place. I put it on myself, so yeah. Now to add the alcohol, like the next agar plate, and it's gonna be and it's gonna be filled with the homemade hand sanitizer I made. Okay, this is the 60% alcohol homemade hand sanitizer. Use the 5 milliliter pipette to get some homemade hand sanitizer. Okay, that's good. I already labeled it, so I can tell which is which. 60 going to be here. Yes. Oh, no, wait, oops, I made a mistake. Oh no. It's gonna be okay, I hope. Oof. This is not good. Okay, time to do my final spread plate. Spreading it with this. Okay, that's good. Now I'm going to use, let's see, according to this plate, I'm going to use 60% homemade alcohol hand sanitizer. Gently make the well. Okay, so far it's good, so good. 70% alcohol. Here it comes. Okay. I made the well for the 70% homemade hand sanitizer. I forgot to say alcohol. Whoops. Now finally the 80%. Alcohol. Homemade hand sanitizer. Okay. Here it comes. And stab. Done. And voila. Perfect. Now I'm going to wait for the result to come. The time that I finish the experiment is March 21st, 1:15. So I have to wait for 24 to 48 hours. This is Gajeena Warren signing off for now. Here's my results. There's fungus that's between the water and the water plus soap section. There's only a few colonies. The Streptococcus aureus, and the orange one I believe it's not E. coli. It's in my it's out in my tip of the tongue. But I know there's lots of Streptococcus aureus here in the water section, and mostly contamination. Store-bought hand sanitizer. Mostly contamination, and one Streptococcus aureus. For the one that is a homemade hand sanitizer, there's no colonies. I don't know what happened. Maybe it's because the wells overflowed hand sanitizer, causing the colonies to be destroyed, or the saline is not enough to produce some colonies. So I'm going to use a cup of dirt plus water in a new agar plate. Yeah, this is going to be water and handmade sanitizer and water plus soap. Okay, let's do this. Going to put this here. Gonna have some soap. There. This is 80% homemade, 80% alcohol of homemade hand sanitizer. Oof. Okay, that's a failure. Now for the 70% alcohol of homemade hand sanitizer. And finally, the 60% alcohol for homemade hand sanitizer. There. Good. Now I have to wait for 24 to 48 hours or more to see if there's colonies exist. And to see if the hand sanitizers, the homemade ones, the positive controls, and the other 2 positive controls, 1 negative controls, can destroy any of the colonies. Till then, signing off. Okay, apparently, well, my results came in. Okay. Apparently I have 1 successful well. Wait, make it 2. But it's apparently because there's a fungus within the well. Yeah. Oh my gosh, I punctured 3 holes. 4. Water plus soap. Yikes. Yeah, this is water for the well. For the control. Oh man. There's no well for all 3 of them, the homemade hand sanitizer, just white fuzzy fungus. Dang it. Guess all my trials are complete failures.